Welcome to AP Biology. Today's video is going to be on non-Mendelian traits. So our first example of non-Mendelian traits is what we call codominance. Co means together, and dominance is dominance. And so when we have codominance, that means these two traits are going to be together dominant. So that means they're both going to be expressed in the offspring. So what do we mean by that? Well, if you have a brown horse and a white horse and you mate them together, you get a brown and white horse. Okay, so we see both of the parental alleles in the offspring. If I mated a white and a brown cow, I'd get a brown and white cow. If I mated a black and a white chicken, you would expect the chicken to come out black and white. If I mated a red and a white flower, the resulting flowers would be red and white. So those are examples of codominance. This is a pair of codominant heterozygotes. Not really, but it's a cool picture. All right, so what is incomplete dominance? So incomplete dominance means neither trait is going to be dominant, and so we end up with a blend of the two traits. So when I mate a rose that's red and a rose that's white, I end up with pink roses. So I get a blend of the two. So neither of the traits of the parents are dominant. Snapdragons do the same thing. If you mate a the reddish purple one with a white one, you end up with pink offspring. Andalusian fowl, if you mate a white Andalusian with a black Andalusian, you end up with kind of a grayish colored Andalusian. Palominos, horse breed, if I mate a brown and a white Palomino together, I end up getting the tan Palomino. So the difference between those two, incomplete means we get the blend, red and white makes pink, and codominance means we get both of the traits in the offspring. So we get the red trait and the white trait in the offspring. So what happened here? This is an interesting one. This is kind of a famous cat named Zeus, and Zeus actually formed from two separate eggs that fused together during development. And so this was supposed to be one sibling and this was supposed to be the other, and they just made one cat. Another form of non-Mendelian genetics is what we call X-linked traits. So X-linked traits are going to be traits that are found on the X chromosome. We have about 55 genes on the Y chromosome, and the Y chromosome is called a Y because this is what it looks like. It looks like a little squatty Y. Those 55 genes, most of those are involved in turning you into a male. So there's not too many genes on the Y chromosome that are necessary for survival. Otherwise, all females are dead because they don't have Y chromosomes. So if there was anything on there that was needed for survival, females could not survive because they have two X chromosomes. And so mostly it's just turn me into a male type genes. The X chromosome has 900 genes, many more genes than the Y chromosome. And these are all involved with many factors of your normal survival. Okay, so the X chromosome is crucial for survival. The Y chromosome really is not. It just converts you at a certain point in development from a female into a male. We all start off female during the beginning of development, and then at about 9-10 weeks, the Y chromosome kicks in and starts converting the female into a male. So how do these traits get passed on? Well, it's a little bit different because now you are dealing with, instead of autosomes, you're dealing with sex chromosomes. And so the Y is not going to be the same as the X. So if you have a female donating an X and a male donating a Y, these are always going to end up male, and these are always going to end up female. The problem is, because males only have one X chromosome, they don't have a backup. So in a female, if I get a bad X, I can have a good X that will cover for my bad X. In a male, if I get a bad X, I'm going to have a bad trait expressed. And so we tend to see these traits that are on the X chromosome much more common in males than females. For example, color blindness is present in about a little less than 10% of males. It's around 7-8% of males. In females, it's about 1 in 250 females. So it's really rare in females. It's like 1 in 15 males and 1 in 250 females. So it's really rare in females and fairly common in males. This is probably the most common X-linked problem that males have. This is a normal color spectrum, but if you're missing a couple of those genes for making your colored cones, you're not going to be able to see in the normal color spectrum. So you tend to see blue and yellow, and that's about the only colors you see very well. Red and green and purple all kind of look the same to you, especially red and green. If you notice down here, this color and this color kind of look the same. So you don't really distinguish those two colors at all. This is a color blindness test. So people who have normal color vision can see the 42 pretty easily. And people who do not have normal color vision just see a whole bunch of spots. I always think this is interesting. This is how we observe, if you have normal color vision, the colors of these two teams, the red team and the green team. If you had color blindness, this is how you would observe them. It's quite a bit different. Notice you can still see the blues and the yellows, but all the greens and the reds pretty much look the same to you. They kind of look muddy. Another type of non-Mendelian genetics is what we call multiple alleles. This is where we have more than 2 alleles that are common in the population. In this particular case, this is human blood type. We have 3 common alleles. You have the A allele, the B allele, and the type O blood, which is not really an allele. It's like the non-existence of an antigen. So there's a few different things that can happen with blood typing. If you have a A gene, you're going to produce A proteins on the surface of your blood cells. And if you have a B gene, you're going to produce B proteins on the surface of your blood cells. And if you have an A and a B gene, you're going to produce A and B proteins on the surface of your blood cells. And if you have recessive defective genes for both those, then you produce no proteins on the surface of your blood cells. It also means that you're going to produce different antibodies in your blood. People who are type A produce B antibodies. People who are type B produce A antibodies. And people who are AB don't produce antibodies against either of those. And people who are O produce antibodies against both. Now, Because of this arrangement, different people can donate to different other people. So somebody who's type O can donate to really anybody. People who are type A can donate to A and AB. People who are B can donate to B and AB. And people who are AB could only donate to other people who are AB. That's why we call type O the universal donor and type AB the universal recipient. And this is what happens if you give them the wrong type of blood. Because the antibodies identify and attack the antigens on the surface, blood tends to clump up. Okay? And so if I gave a type O person A blood, clumps. B blood, clumps. AB blood, clumps. The only thing an O person can get is O. They have both the different antibodies, and so when they see these other types of blood, they attack them and clump them up. So you can see type O can only receive type O, but type O can donate to everybody. AB can receive from everybody, okay, but they can only donate to AB. This is another example of multiple alleles, the color of rabbits. So you have black, chinchillian, Himalayan, and albino. When you have two dominant traits for black, you end up black. If you have one dominant trait for black, you end up black. Okay? If I have a chinchillian and a black, I'm black. But if I have two chinchillians, then I'm chinchillian. If I have a chinchillian and a Himalayan, I end up chinchillian. Each one of these dominates the other one. Okay? This means I have two recessive traits for both. Neither one of those produces any coloration, and so I end up albino.